Hello everyone, welcome to biology class. Today our lesson is about protein synthesis. So we're going to talk about the process of transcription. Before starting our lesson, I want to introduce you some history. It started in 1902 with the British physician Archibald Garrett, and he noticed that each gene synthesizes the specific enzyme, which catalyzes the specific chemical reaction in the cell. So, Archibald Garrett suggested that symptoms of inherited disease are caused by an inability to make a specific enzyme. For example, alkaptonuria, it's the disease of black urine. It means that because of chemical alkapton, which gives a dark color to urine, the color of urine becomes dark, especially a black one. So, also Archibald Garrett introduced one gene and one enzyme hypothesis. It means that each gene dictates the production of a specific enzyme. In 1958, George Beadle and Edward Tatum from the Stanford University proved this hypothesis, one gene and one enzyme, and they won the Nobel Prize. Okay, let's start it. Okay. Here you can see the DNA and RNA. So before the starting our lesson, you need to know these two specific terms, DNA and RNA. So we know about the gene located in the nucleus in the cell, and in the genes we have the DNA and RNA. DNA is deoxyribonucleic acid, and it provides instructions for protein synthesis. But genes do not build the protein directly, so there is a bridge between DNA and the protein synthesis, and we call it mRNA, messenger ribonucleic acid. Here, in the structure, we have differences in DNA and RNA. Here you can see they are similar, but DNA is two-stranded. It means that they are double-stranded. It's the one helix, and here the next second helix. But RNA is only one-stranded. And there is a difference in their nucleotides. Here in DNA we have thymine, cytosine, adenine, and guanine, but in RNA we have uracil instead of thymine, and the cytosine, adenine, and guanine as well. Here you can see the differences in this table. We see its structure. Yes, DNA double-stranded, but RNA it's only single-stranded. Sugar here, deoxyribose sugar, but in RNA we see ribose sugar. In DNA here the bases adenine complementary with thymine and cytosine complementary with guanine. But in RNA we see adenine complementary with uracil, cytosine complementary with guanine. Also in their location, DNA always located in the nucleus, but RNA located in the nucleus and in the cytoplasm also. And there's a difference in their function. Like DNA is always about genetic storage, but the RNA it's about the genetic transmission. Here are the types. RNA has three types. It's messenger RNA, transfer RNA, and ribosomal RNA, but DNA has only two types. It's genomic or mitochondrial. And others, here you can see their nucleotides and their difference. Ribose sugar, deoxyribose sugar, and their nucleotides. Okay, next one. Here, so we say that DNA has the information for cells to follow, but however, cells do not understand the DNA language. So that's why it's a problem. In order to solve this problem, the DNA code must be translated in order to cells to get the instructions. That's why DNA firstly copied in the nucleus first. We know this process as a DNA replication. After that, the message is translated in the cytoplasm at the ribosomes. So firstly, the process of transcription. After that, the process of translation. Here, definitions, we can see you have the three major processes. The first one is central dogma, transcription, and translation. The central dogma is a process that describes the basic framework of how the genetic information flows from DNA sequence to a protein. So firstly, DNA transcribed into RNA, mRNA. After that, mRNA translated into protein. And after this process, the polypeptide chain is formed. In the transcription, they get the information, genetic information from the DNA, and it formed or changed it into mRNA. In the translation, the mRNA translated in the form of proteins as amino acids. So we're gonna talk about the transcription. Yeah, here also, before the transcription, we have the replication process. You must know about it. After the DNA replicates itself, the next stage is transcription. Transcription is the process of synthesis of RNA from DNA. In other words, it copies the genetic information from DNA into RNA. So here we have the 3 stages of transcription. First one is initiation, elongation, and termination. But the 3 stages are common all living organisms, but there's a several difference between prokaryotic cells and eukaryotic cells. Yeah, if you don't know about them, please you need to read about them, that the prokaryotic cells, they don't have nucleus, eukaryotic cells, and they have nucleus. That's why there is a difference in the processes. So first is transcription, begins with the process of initiation. Yeah, as you see here, we can see the RNA polymerase. RNA polymerase is an enzyme which helps to start this process. It's an enzyme that catalyzes the process and gives us opportunity to speed up the reaction. Here, we have the promoter. Also, recognizing the promoter is essential for RNA polymerase because it helps RNA polymerase to start the process at the right place. Here we can see the coding strand of DNA, double helix. It starts from the 3' and 5 end. And here we can see the termination site, but before it will occur in the initiation. Here, next one is the process of elongation. In the elongation, you can see that the messenger RNA transfers information from DNA to protein. Yeah, it's synthesized from the template strand of DNA, here the template strand, and it begins to start copy the genetic information in the form of messenger RNA. Here in the yellow one or in the orange one, you can see the RNA, it's the messenger RNA. Here you can see the strand of DNA. Nucleotides are added to the 3 end of the growing RNA strand. Here you can see the nucleotides, and they come together, complementary strand, and they form a messenger RNA. The third one is termination. Termination, it means the end of this process. But in eukaryotes, RNA polymerase transcribes a sequence on DNA called polyadenylation signal. It means that polyadenylation signal consists of 6 nucleotides. It's A, uracil A, and 3A. It means that this signal gives the mRNA to end this process. So RNA polymerase recognize the signal, polyadenylation signal, and it ends the process. So finally, the product is mRNA. mRNA transcript is built in the end. Here we can see the video of how DNA changed from its information.
Here is a cell, the basic unit of all living tissue.
So,
in most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription.
Other nucleotides, and after this process we have made a messenger RNA. So So we have made all the thing, and next I think that it's the time to do tasks. Okay, I think that everything is clear. So if you understand the topic, we can start doing tasks. Here's the protein synthesis tasks. The first one is transcribe the following DNA sequence into mRNA. Yeah. So here we can see the mRNA. It's A, T, C, G, T, C, C, A, and we need to convert it to mRNA. So here we can see the matching DNA and RNA. We know that adenine complementary with uracil, because in RNA we don't have thymine. Thymine complementary with adenine, and cytosine with guanine with cytosine. Yeah. Why here uracil? Because in RNA we don't have thymine. That's why they will be complementary with uracil. So adenine here we will be complementary with uracil. Uracil. Thymine will be complementary with adenine. Cytosine will be complementary with guanine. Guanine will be complementary with cytosine. Thymine complementary with adenine. Cytosine complementary with guanine. Here also guanine. Here also uracil and uracil. Okay, next one. Here the following one. We need to transcribe the following DNA. Okay, here we can see that thymine complementary with adenine. Adenine complementary with uracil, guanine with cytosine, cytosine with guanine, adenine complementary with thymine, guanine complementary with cytosine, guanine complementary with cytosine, thymine complementary with adenine, and here also adenine. Okay, next one. Here also DNA strand. Here will be adenine complementary with uracil, cytosine guanine, guanine cytosine, cytosine guanine, and guanine cytosine. Here also guanine, and here adenine complementary with uracil, guanine complementary with cytosine, adenine complementary uracil, guanine complementary with cytosine, cytosine complementary with guanine. Here thymine complementary with adenine, adenine complementary with uracil, guanine complementary with cytosine. Okay, here. And here we have the two questions. First one is, what is made at the end of transcription? Yeah, we said that DNA copied into mRNA, so the answer will be mRNA. The B1. DNA, it's not the answer. Protein chain, it's not the answer. And the tRNA, it's not the answer. Next question, what is made at the end of the translation? So we have the mRNA, and after the translation, mRNA changed into protein or amino acid. Amino acid is monomer, protein is a polymer, that's why it's the same thing. Or we can say polypeptide change. Why polypeptide change? Because the bond between amino acids is a peptide bond. That's why there are several bonds. That's why it will be the polypeptide change. So answer is the C, yeah? Protein change. Not tRNA, because tRNA just helps. Not mRNA, because it's the product after the transcription. Not DNA. So answer is protein change only. Okay, I think the tasks are clear. So if you have any questions, please write down in the comments below. And that's the end of our lesson. So thank you everyone. Goodbye.